These Sertoli cells are also present inside the seminiferous tubules, and these Sertoli cells are actually going to play a crucial role in the maturation of this round spermatid into spermatozoa. These spermatogonium are present here. After this spermatogonium, you will find some another kind of cell. Those are known as. Hello Bandhagan, Namaskar and welcome back to my YouTube channel Sahil Educations, and it is the second episode of the chapter Human Reproduction. Today we'll be discussing about the process that is known as spermatogenesis. A very common process is always going in your body, in the male body, basically after adolescence or after puberty, not exactly adolescence. Okay. Before starting the process of spermatogenesis, we just recall the structure of testis or anatomy of testis. The last layer of this testis is the tunica albuginea, and this tunica albuginea engulfs inside the testicles and it makes a structure named as septa. And in between two septa you have understood that a lobule like structure forms, and the number of lobule in a single testicle is approximately 15 to 20, and each and every testicle contains 2 to 3 seminiferous tubules. Okay, I'm coming upon this seminiferous tubule later on. Then this seminiferous tubule is basically the structure present inside the testicle in which the sperms are being formed and matured. After maturation, the sperm enters into the retitestis. After retitestis, the sperms enters into the vasa efferentia, and then sperms enters into this epididymis. Epididymis has 3 parts. This one is the head part, this one is the body, and this one is the tail region of the epididymis. After epididymis, sperms enters into the vas deferens. So let's discuss about this seminiferous tubule. As you people know that tubule must be a tubular structure. If you cut this tubule and see under the microscope, you will see a circular structure now. This one is the peripheral region and this one is the lumen. This one is the peripheral region of the tubule. Peripheral region. And this is basically the lumen of the tubule. And the same happens with your blood vessels. In case of blood vessels as well, the peripheral muscular layer is present and the lumen is present. In the lumen region, basically the blood passes through. Okay, so all these things are basically this structure is about the seminiferous tubule. The tubular structure actually looks like this. And orientation of this tubular structure is like the peripheral region is present and a lumen is present in a tubule. Let's talk about a cross section of this testis. If you just cut this section of testis, what will you see? You'll see a block, square or rectangular block under a microscope, and then the seminiferous tubules will look like this. So these are your seminiferous tubules, circular in structure. As the tubule has been cut in a cross section, a transverse section, it's not a longitudinal section. So after these seminiferous tubules, what are the structures present outside this seminiferous tubule? You will see some blood vessels, definitely the arteries and veins. Like this, just to keep your cells present in the testicle healthy, so that the blood can blood can provide the nutrient, provide the glucose, provide the oxygen to the cells present in this structure. And apart from these blood vessels, you can see some another structures, another kind of cell, which is known as Leydig cells, And as the Leydig cell is present in the interstitium structure, interstitium is basically the region present outside this seminiferous tubule. Whatever region is present outside this tubular part is known as interstitium. And if the actually these Leydig cells are present in the interstitium, that is why these cells are known as Leydig cells of interstitium. Or you can call it interstitial Leydig cells. Okay, so basically this structure is present in this microscopic or histological region, or these kind of structures are present in the cross section of a testis. Now talking about the function of these Leydig cells, these Leydig cells perform the production of testosterone in male body. Testosterone is produced by the interstitial cells of Leydig. Okay, and now look inside the, now we are going to look inside the seminiferous tubule. When we study the structure of seminiferous tubule, the cross-sectional structure of seminiferous tubule, you will firstly find a basal membrane like structure. The first layer which surrounds the seminiferous tubule is the basal membrane. We call it basal membrane. And this membrane is formed of myoid cells. MYOID, myoid cells. Okay, after this basal membrane you will find another cells which are basically the stem cells, which are known as spermatogonium, spermatogonial cells or spermatogonium. Okay, so these spermatogonium are present here. After this spermatogonium you will find some another kind of cell, those are known as primary spermatocyte. After this primary spermatocyte you will find another line of cells which are known as secondary spermatocyte. After this secondary spermatocyte you will find round spermatid, and these round spermatids will get matured, having a tail like structure and an acrosome at the head part. This is known as spermatozoa or sperm. This formation or this maturation of round spermatid into the spermatozoa is known as spermiogenesis, and that spermiogenesis is being performed, or some kind of cells are going to be involved in this process, and the cell's name is the Sertoli cells. These are known as Sertoli cells. These Sertoli cells are also present inside the seminiferous tubules, and these Sertoli cells are actually going to play a crucial role in the maturation of this round spermatid into spermatozoa. And the conversion or maturation of spermatozoa from the round spermatid is known as spermiogenesis. Remember this, you may get some question in your NEET exam on this topic, spermiogenesis. So spermiogenesis is maturation of spermatid into spermatozoa. sperm. Okay, now there is another process which is known as spermiation. Remember this, spermiation is the process of release of this embedded sperm upon the Sertoli cells. Release of this embedded sperm cells from the Sertoli cells into the lumen of seminiferous tubule is known as spermiation. NCERT has this definition written in the chapter spermiation. So spermiation is basically release of sperm into the lumen of seminiferous tubule from the Sertoli cells. Okay, so there is a difference between spermiogenesis and spermiation. Now discuss about the spermatogenesis in details. See, what you have seen under this basal membrane made of myoid cell, you have seen the spermatogonium. And we have taught that we have already understood that spermatogonium is a kind of stem cell that divides by mitosis, and after mitosis it can produce some another kind of cell as well as some spermatogonium. So let's say that this spermatogonium is going to divide by mitosis. And is producing another kind of cell which is known as primary spermatocyte. Okay, after this primary spermatocyte, it will again undergo mitotic division. It will now proceed for the meiotic division. So it is meiosis 1. After meiosis 1, you will get 2 cells, and those cells are equal in size, equal in surface volume ratio, equal in every aspect. Primary spermatocytes undergoes meiosis 1 and it produces 2 cells of equal size, equal length, equal in every aspect, and those cells are known as secondary spermatocytes. After this, secondary spermatocytes will now again go for the completion of meiosis, that is meiosis 2. After meiosis 2, these 2 secondary spermatocytes will produce now a total sum of 4 round cells. These 4 round cells are just embedded in the Sertoli cells. Look, if this is your Sertoli cells, the cells embed like this, and these cells are known as spermatid, round spermatid. Okay, let's talk about the ploidy now. If we talk about the ploidy, spermatogonium is 2n. If it goes through mitotic process, then it will again produce the cells which have 2n number of chromosome. And then meiosis 1 is a reductional division, we all know this, and meiosis 1 will give rise to n number of chromosome containing cells, and those are the secondary spermatocytes. After this, the meiosis 2 is being completed. Again, it is an equational division. Then it will also contain the n number of chromosome in every spermatid, every round spermatid you can say. After these round spermatids are formed and they are embedded in the Sertoli cells, they will undergo maturation, and that maturation process is known as Spermiogenesis. So in this process, basically these cells will get its proper shape, which contains a long tail-like structure, a short neck-like structure, and a head, and one head here. These cells are yet embedded into the Sertoli cells. These cells are yet embedded into the Sertoli cells. They have not been released from the Sertoli cell yet. Okay. So these are the basically matured spermatozoa. If they have the cap-like structure which is known as acrosome, and these acrosomes are being formed by the Golgi apparatus. These acrosomes are being formed by the Golgi apparatus present in these Sertoli cells. These Golgi apparatuses will produce acrosome granules, and those acrosome granules will be accumulated, and they just form a sac-like structure which is known as acrosome. Now the spermatids are matured, and these are known as spermatozoa. And these spermatozoa have a ploidy containing n number of chromosomes. So you have produced your sperms. So this is all about the process of spermatogenesis. Now, if these sperms get released from these Sertoli cells and comes into the lumen to go inside the retitestis, inside the vasa efferentia, inside the epididymis, then this process, just releasing these spermatozoa from the Sertoli cell into the lumen, this process is known as spermiation. Hope you have understood the concept of spermatogenesis as well as the histological structure of testis. Now we will see you in the next video. Thank you very much.